Okay, we're talking about enzymes here, which are an especially important group of organic molecules because they conduct almost all biological processes. Now, enzymes are proteins, for the most part, of sequences of amino acids. However, some RNA and nucleic acid molecules have been found to conduct these reactions. Some of the important points we want to get across about enzymes, that they are catalysts. In other words, they conduct these chemical reactions. They take reactants and make products. And the important role they serve is to speed up these chemical reactions inside the cell. Now, they bind to these reactants to form products based on their molecular shape. So there's a very specific shape that they have to bind those reactants. And their activity, how well they work, is affected by temperature and pH, as well as the presence of things we'll investigate called inhibitors. Enzymes make reactions go much more easily because they lower the amount of energy required for a reaction to occur. So let's take a look at an important reaction in our red blood cells in our lungs, where H2CO3, carbonic acid, is converted into CO2 and some water. Now, this reaction is essential because we want to unload this CO2 into our lungs with every exhale that we take. So, as we can see here, without an enzyme present, a certain amount of energy is required to be put into this before that reaction can, as we say, go and form products. Thus, here would be the amount of energy required for that reaction to go. Now, in the presence of an enzyme, carbonic anhydrase, we see a much lower amount of energy is required for the reaction to go. And that, in turn, makes this reaction go much more quickly. An enzyme has to bind that reactant, and we call that a substrate. And so here in this three-dimensional image here, we see an enzyme with its substrate bound inside its active site, which fits in there very specifically. And that substrate, again, is called the reactant in a chemical reaction. So this would be like the carbonic acid we just saw in the previous reaction. Now, when that substrate binds inside the active site of the enzyme, we call that the enzyme-substrate complex. Now, the binding of the substrate to the active site is referred to as a lock and key sort of model, where the substrate has a very specific shape that fits in the active site like a key into a lock. And when that happens, we also see a slight change in the active site as a better fit, called an induced fit, to that substrate. And here you see the enzyme-substrate complex. The reaction is catalyzed. We see here that we've separated these two molecules from each other and formed the products. Now, important to note is the fact that this enzyme goes back to its original shape here and can be used again to catalyze another reaction. Now, this example could easily be the hydrolysis of sucrose by the enzyme sucrase, which breaks up sucrose into its component parts, glucose and fructose. So it's important to understand what kind of things affect the activity of enzymes. The first is denaturation, where an enzyme loses its shape due to some kind of change in environmental condition. There's an unfolding of the enzyme. So we see an enzyme that looks like this suddenly become just a long ribbon of amino acids. And this is usually due to some change in either pH or temperature. And of course, the loss of that shape, which is very important to an enzyme's function, renders it biologically inactive, and that's because we have changed the composition of those active sites. There's no longer the specificity needed to bind a very specific substrate. Now, there are optimal ranges of pH and temperature that all enzymes operate within. For instance, in our bodies, the enzyme pepsin can be found in our stomachs, which in our stomach has a very low pH of around 2, and of course you can see pepsin operates the best in that pH range. Enzymes like amylase and trypsin could not possibly operate in our stomachs because they require much higher pHs of 7 and 8. Okay, another important factor that influences enzyme activity is inhibitors. And inhibitors are molecules that are not the substrate, but that can bind to the enzyme and prevent it from functioning. Now, there are a couple of different kinds of inhibition caused by inhibitors. The first is competitive inhibition. So here we see on this enzyme, again, the very specifically shaped active site, and we see the inhibitor coming in that has a shape that will fit and bind to that active site. But this isn't the substrate for the reaction. And so it prevents the actual substrate from being able to bind to the active site of the enzyme, and thus prevents the enzyme from functioning. Now, in non-competitive inhibition, we see that the inhibitor now binds to another place on the enzyme. And when it binds to that place on the enzyme, suddenly the active site changes its shape as a result of that binding. So you see here this very triangular shape, and then after the binding of the inhibitor, a change in that active site, which of course will prevent the substrate from binding altogether. Now, inhibitors are important tools in the design of medicine. Here again, a very complicated enzyme, dehydroquinone synthase, which is an enzyme involved in amino acid production in bacteria that is not in our bodies ourselves. And we can see a number of things bound to this enzyme. Inorganic ions, organic molecules, things called cofactors and coenzymes. But here is where the actual reactant would go, the substrate. And this, what you see bound here, is the inhibitor, carbophosphonate in yellow. And of course, the binding of the inhibitor prevents this whole enzyme from functioning and creating the amino acid. So if we can design medicine where we place this inhibitor in the presence of these bacteria, they will bind that inhibitor, they will not be able to make their amino acids, and they will not be able to proliferate. And this is how antibiotics are often made.